All right, so this video, it's going to be focused specifically on just glycolysis. Now, glycolysis, as the name implies, that word lyse or suffix lyse just literally means split. So when you think about it, it's glucose splitting in two. So before we get into the logistics here, you just need to know really what is the point. There's a few things here that I like to think about is to, you're basically taking glucose to pyruvate, okay? And glucose is getting oxidized. So you're gonna make some NADH, which will later be used for the electron transport chain. But the key thing here that I need to really emphasize is all cells, all conditions. And what I mean by that is all cells, whether it's prokaryotic or eukaryotic, can do glycolysis. All cells, in terms of oxygen or no oxygen, in terms of the condition standpoint, they're able to do this process. So it's really, really significant for us to understand that you are going to make some ATP. It's substrate level phosphorylation, which if you didn't watch in the last video, it just means that it's a way to make ATP in the sense that you can pull a phosphate off of a substrate, whatever it may be. You're going to push that phosphate on to ADP and make ATP. So yes, you're making it. It's not the most efficient, takes some time, but it is a way to make ATP in the absence of oxygen. So all cells, all conditions takes place outside the mitochondria. So I'm going to show you the couple of animations I have here real quick. So, and again, all this is from Mastering Bio. Feel free to go back and look at this on your own. But
which begins
let's take a quick look here. So right here I have glucose. It's outside the mitochondria. Glucose has 6 carbons, and that's what each one of those black spheres represents. And notice what happens here. You actually have to use some ATP to get this process going. So the analogy used here a lot is that you have to spend money to make money. You have to actually spend some ATP to make some ATP. So I've split glucose right down the middle. It's not called pyruvate just yet. But I got some phosphates here. I have 3 carbon molecules, but the point here is that I got to actually pull some electrons off. I'm going to actually oxidize these 2 molecules. So by the process of oxidation, I'm going to get some NAD plus coming through, pulling electrons off and making some NADH, okay? So this is the energy investment phase. So let's move on to the next part about glycolysis and see what happens here. I'm still down to ATP, so let's see what makes this whole process worth it. So what's going to happen here is NAD plus is going to get reduced to NADH. It's pulling electrons off, okay? I'm going to add another phosphate here, not from ATP, but now I have these 2 3 carbon molecules here. There's a lot of energy here. I'm going to do substrate level phosphorylation right there. I'm going to pull these phosphates off on this and actually get a net gain of 2 ATP. So these phosphates can still be removed. So they get shuffled over, they get hydrolyzed, and then you can actually make 2 more ATPs. So that gives you a net production of 2 ATPs and 2 NADHs. So when you jump back to this, let's kind of take a look at what you should at least be able to understand is the payoff. You get 2 ATPs produced and 2 NADHs. You do make 4, but remember you actually had to use 2 ATPs to get there. So you're making some pyruvate. Glucose is 6 carbons in length. You make 2 pyruvates, which are both 3 carbon in length, okay? Now, if you go back to this, let me show you this quick animation. I'm gonna go and zoom out real quick, and then we can look here. I like this visual a lot, so let's look at glycolysis. This video, it's not showing all the steps, but it kind of gets the point across that, okay, here's glucose, there's a mitochondria back in there. So now we're inside the cell. And I'm going to use those 2 ATPs to get this process going. So adding 2 ATPs, I'm going to phosphorylate it. So now this has been phosphorylated and then it gets split, okay? It's going to get split down the middle. We're just going to focus on one. The other one is going to be doing the exact same thing. But here's NAD+. It picks up the electrons and then it becomes NADH. Okay, so there is going to be your NADH. It's carrying electrons. Notice it has another phosphate. So I'm going to make 2 ATPs from this one, but don't forget there was that other one that we're not showing in this process. So this isn't called pyruvate just yet. I'm going to pull a phosphate here. That's substrate level phosphorylation. I'm going to pull this phosphate here, and that becomes ATP, and that is substrate level phosphorylation. So really, when you think about the whole process of cell respiration, we got to talk a little bit about the reactions. So this is glycolysis, right? So all cells, all conditions can do this. Now, glucose will eventually make pyruvate, and it takes really 10 steps to get here. Now, each one of these has their own specific enzyme, so this is what I'm kind of showing you here. I'm making these weird different shapes to represent each enzyme. Maybe this one looks something like this. I mean, it doesn't necessarily look like that, but it's just getting the point that each one of these requires their own enzyme. When you take biochemistry in college, you'll for sure have to memorize the names of each one of these enzymes and kind of what's going on here. But I do find this important for you guys to understand. Glucose, let's look at step one. This is glucose right here. Glucose, to make this first intermediate, is an exergonic reaction, right? This delta G value is negative 16 kilojoules per mole, okay? Now, you do not have to memorize these by any stretch of the imagination, but it's kind of interesting that when you look at it, this is the enzyme hexokinase. So I would call this guy hexokinase. Hexokinase is involved right here. That's the enzyme. It's going to take glucose and make your glucose 6-phosphate. So do you have to memorize this? Absolutely not. What I'm trying to emphasize here is that all of these cells are going to share some similar enzymes, meaning if we looked at our hexokinase enzyme here compared to any other mammal, it's going to be nearly identical. All cells are going to have a form of hexokinase. And in terms of similarity and the function of what they do, it's very, very similar. It's going to take glucose and make that glucose 6-phosphate. They're each going to have their own second enzyme here, or glucose phosphate isomerase. I'm not even going to try to write that over here, but this is called glucose phosphate isomerase. So it's going to take that glucose 6-phosphate and form what's called isomerase right there. So, or excuse me, fructose 6-phosphate. But I do want to take a second and look at this. Let's look at step number 4. And I want you to take a look at the delta G value. So this is a positive value. So that means going from here to here, it's a positive delta G. This one was negative. So I want you to at least understand this. Going from glucose all the way to pyruvate, it takes enzymes involved at each step, okay? 10 different enzymes. Now, some of them are exergonic, some of them are endergonic. But the overall, this is what we look at, this is really what we care about. Actually taking glucose and forming pyruvate, the overall delta G value is negative 73.3 kilojoules per mole. So is this whole thing spontaneous? Yes, in the presence of the enzyme actually going from glucose to pyruvate, you make some ATP at a couple of different steps. But notice that this is the phase where you actually had to add in ATP. So like if you look at step number 7, yeah, it's exergonic because you're actually coupling it with some ATP. But regardless, you just need to understand what is glycolysis. You're taking glucose, which is 6 carbons, you're making 2 3-carbon pyruvate molecules, and you get a net production of 2 ATPs. Yes, you had to spend a little bit of ATP to get this process going, but you had a net production of 2 because you produced 4, but you had to spend 2. So you make some ATP via substrate level phosphorylation, and you also made some NADH. NADH will be used for the electron transport chain in the later videos. So I hope that is somewhat helpful. The biggest thing that I need to emphasize here when you look at it, all cells can do this. And when you think about it, it makes sense because no mitochondria is needed. Glycolysis, no oxygen is needed. So glucose gets quickly converted into pyruvate. You make some ATP and you make some NADH. Right? So that is glycolysis. So just jumping back, summarizing this whole thing up, let's just recap looking at this video. If I replayed this, again, you spent 2 ATPs to get this thing going. You're going to remove electrons off of this guy to make some NADH, and then you're going to make a little bit of ATP. So there's NAD+, picking up electrons, making NADH, picking up electrons, making NADH, and then you're going to pull these phosphates off to make some ATP. So you can see it here nice and clearly. You make some 2 ATPs, this phosphate gets shuffled around, but the key thing is that you make 2 ATPs, 2 NADHs in glycolysis.